So welcome to this masterclass from Cambridge Leadership College. My name is Warwick Bailey. I'm one of the Z coaches at the college. This evening, in this masterclass, we're looking at a very recent paper, 2016. It's the multiple choice paper for AS level biology. And there's 40 questions. We're going to be pushed to do all of those in the time that we've got. So what we'll do is we'll focus on the questions that the examiner identified as the ones that the students needed to particularly focus on, and also make a particular focus in this masterclass on biochemistry. For those students who are doing CIE chemistry as well as biology, there's quite a lot of overlap between the studies of biochemical molecules that you study in biology and the biochemistry part of the chemistry syllabus as well. So if we make an emphasis on some of the macromolecules that you cover in biology, that satisfies some of the chemistry study as well. And with these multiple choice papers, they often start with questions related to size, and that could be a question on microscopy, looking at magnification or something like that. And so before we do a dive into this paper, which actually, like the others, starts with lots of questions about size, I just wanted to review some of your knowledge about size before we start getting into answering some of these questions. So let me just try and scroll up this paper here. There we go. Okay, good. All right. So I'm just going to use this little bit of space down here to think about size for a moment. And where's my, there we go, okay. So on your metric scale, of course, you've got your meters, and then you're stepping down in thousandths, aren't you, from meters? So millimeters, you've stepped down 10 to the minus 3. The next step down, 10 to the minus 6, is micrometers. Not writing very well this evening. Next step down from that is nanometers, 10 to the minus 9. And you can see how this keeps going. And then you get down to picometers, that's 10 to the minus 12. We could keep going. We're not going to do that because that is really getting too small for biology. But so that's the range that you need to know about, stepping down from meters. And what I wanted to do was just to take the range that you're most working with cellular biology, which is about millimeters through micrometers through nanometers, and then just put on here some familiar concepts from cell biology or from chemistry, actually, to give us some sense of the ranges of the sizes that you're dealing with. So start off with a bit of chemistry then. The size of an atom is going to be, that's going to be in this range here, about 0.1 of a nanometer. This is the nanometer column here. This is the micrometer column there. All right. So atoms about 0.1 maybe to about 0.2 for nanometers, and then bond lengths between atoms. Well, for reasons that you study in chemistry, the bond length in atoms, between atoms in molecules, is similar to these kinds of values that we had for atoms themselves. So bond lengths about 0.1 to 0.2 nanometers. And then coming in, so that gives you a sense for atoms and how atoms are bonded in small molecules. And then when you start to get into the realm of biology, let's say if we had a hemoglobin subunit. Okay, so that's, I suppose you think of that as a single peptide chain, a hemoglobin subunit, an enzyme, something like that. So you're about 5 nanometers for one of the subunits of hemoglobin. Of course, hemoglobin is composed of 4 subunits, so it'll be a bit bigger than the individual one there. Coming a little bit larger than an individual protein then, a virus, that's going to be about 100 nanometers. Okay, so quite substantially bigger than just a protein chain. And then you can start to say, well, this is now starting to get towards the micrometer scale. So 100 nanometers, 0.1 micrometers. Remember, these are stepping down in units, or stepping down by 1,000 for each of the metric prefixes there: milli, micro, nano, and pico. Okay, viruses, very small. Bacteria, that's going to be about 1,000 nanometers, or you could say 1 micrometer. Then looking at some of the organelles within a cell, had a mitochondria. That's going to be about 3 micrometers. Bigger than that, a chloroplast. That's going to be about 10 micrometers. Nucleus, got that slightly out of order, about 6 micrometers. And for a eukaryotic cell, people who've come to my master classes before know that my writing with these pens is not very good, sorry about that. So the whole cell, well, cell sizes vary quite a lot. So a range of about 25 to 100 micrometers for a cell. So we've gone there up from an atom through to molecules, through to protein chains, through to more recognizable biology elements here: virus, bacteria, these organelles, and right up to the eukaryotic cell itself. Now, for human unaided sight, just good eyesight for humans unaided, then a human can see about 100 micrometers, or that's 0.1 of a millimeter. Okay, so the largest cell is just at the range of discrimination of human vision. All right. But then for aided human vision, the light microscope is using visible light, and that's in the range of 400 to 700 nanometers for the wavelength of visible light. And the resolution for light microscopes is going to be about half the shortest wavelength here. So a light microscope has a resolution of about 200 nanometers. Now, if you look to see on this scale here, then the light microscope, and that's its best resolution, about 200 nanometers, is going to be able to start to pick out about that point there, isn't it? Okay, so larger than a virus, but more discriminating, high resolution than a bacteria. So that's the light microscope, and of course, have we got that right? Yes, I have. That's right. So the light microscope is about here. But there's going to be a lot of detail that we're not going to be able to see then for these organelles. You see, these are quite close to the limit of the light microscope. So to get good resolution for the fine structure of organelles, the light microscope isn't really sufficient. So you're going to need the electron microscope. And that has a resolution of about 5 nanometers, so very substantially better resolution than the light microscope. And so the electron microscope is going to allow us to see a very fine resolution then for the organelles, that you can distinguish the organelles with the light microscope, but to see the structure, the fine structure of the organelles, then the electron microscope is going to be needed. So that's sort of quick run through distances in chemistry and in cellular biology. and looking at the kind of resolution that you can get with a light microscope and an electron microscope is going to be the key in to some of these early questions about size that typically occur at the start of these multiple choice papers. Okay, so here you go then. So we're now talking about organelles, all right? And we said down here that really to see this fine structure of the organelles, then really to get the fine structure, you're going to be using the electron microscope, because the fine structure is not really going to be resolved with the light microscope. So there's a question up here about which of these organelles are going to be clearly visible when you're using the light microscope. Well, these lysozymes are much, much smaller than organelles, so we're not going to be able to see those. The mitochondria and the chloroplasts, well, you can see there that they're sort of at the just about within the range of the light microscope. The chloroplasts are larger than the mitochondria. So if there's any of these that are going to be clearly visible under the light microscope, then we'd have to opt for chloroplasts rather than mitochondria. And for the endoplasmic reticulum, can't say that word very well, the ER, that's actually, isn't it, that for if you can, for the nucleus, that is part of the outer membrane of the nucleus there. So it's actually, it's going to be much smaller than the nucleus. And the nucleus, again, is something which we can pick out with a light microscope, but we can't get very much resolution about the fine structure about it. So all of these then are, that's not discernible with a light microscope, and these aren't going to be clearly visible. So the one that is clearly visible with the highest power of the light microscope is going to be the chloroplast. And so when you look at that table there, then this row D, where we don't have that, we don't have that, we don't have that, but we do have that, that's the one that's going to be clearly visible under the highest power of the light microscope. Okay, so these questions now continue with the theme of size. You can see actually as we get through here as well, there's more questions about size. So all these early questions about size. And this is now looking at not a light microscope, but an electron microscope. Okay, so we've got this much larger magnification here. We had a magnification of 400, which was the maximum with the light microscope. We've now got much higher magnification with the electron microscope. And we're told, aren't we, that there's this measurement here, which is the widest part of the cell. And so what you actually have to do in the exam, you have to go and measure that with your ruler. And when I measured that, that came out at 3 centimeters for me. And so you have to work with the magnification is equal to the measured size over the actual size. Now, we're told up here, aren't we, that we've got a magnification that's times 1,500, and the measured size we just had at 3 centimeters. So plugging all those in, our magnification was by times 1,500. The measured size was 3, and we really need to know here what the actual size is. So just rearrange that. The actual size of this largest width of this cell here is 3 over 1,500. I've just rearranged the magnification and the actual elements in that equation. And working that through, you end up with 2 times 10 to the minus 3. And we had, this was in centimeters, wasn't it? So the actual size is 2 times 10 to the minus 3 centimeters. Well, how many of those is going to fit into a 1 centimeter length? Well, you've got to divide 1 centimeter by each of these. That's finding out how many of those fit into 1 centimeter length, and you get that answer there. Okay, I just want to skip down to this next sort of size question here while we're working on the topic of size. And you can see here that we've started talking about the size of a molecule, okay, glucose molecule, and it has approximate size of 2.4 nanometers. Now, if you just scan back up to this chart of sizes that we had up here, well, each atom we had here was about 0.1 nanometers, and well, glucose is, I mean, it's not that big a molecule. It's a monosaccharide, isn't it? So you've got C6H6, sorry, H12, start again, C6H12O6 for your molecule of glucose. So the glucose is 2.4 nanometers, but all these other examples here are much, much larger, aren't they, than a single molecule of glucose. So let's take the smallest one then, that you've got a polysaccharide here. So that's going to be many glucose, maybe thousands of glucose molecules joined together. And then you've got this hemoglobin. We don't know if that's a hemoglobin unit or 4 hemoglobin units joined together. But for the hemoglobin unit, we had the subunit was about 5 nanometers. So that those protein chains are going to be too large to fit through the partially permeable membrane. So those can't fit through. The glycogen is a polysaccharide. It's many of these glucose molecules joined together, so that can't fit through. Bacteria, we had those, didn't we, about 1,000 nanometers or 1 micrometer. And these ribosomes, that's an example of an organelle within the cell. And when we looked at some of the organelles earlier on, those organelles were in the range of, I suppose, 1 to 10 micrometers. So much, much larger that can fit through these pores in the membrane. Okay, so this is just really a check of your understanding of the relative sizes of these chemical and biochemical and biological entities, and all of those are going to be much too large to fit through the size of the pores in the partially permeable membrane. The only thing that's going to be fitting through a pore or a hole of 2.4 nanometers is going to be fairly small molecules like a monosaccharide or something like that. So which of the following could pass through the pores? Well, that's a slightly kind of tricky question, but the answer there is none of those. Okay, they're all proteins or complete cells in the case of bacteria, or they're polysaccharides. They're all too large to fit through these small holes in the partially permeable membrane. Okay, let's run down to this question here. Okay, and you've got some tests here for biological molecules. And you're given 2 solutions. Solution 1 contains starch and sucrose, and the other solution contains glucose and a protein. We don't know what the protein is. And you need to have in your head really here that the familiar way of writing glucose is as a ring, but the ring is formed from a straight chain form, and if you look at the straight chain form of glucose, at one end there is that chemical functional group, which is an aldehyde. And then you've got OH groups as you go along the carbon chain, like this. This is glucose. And when glucose forms its ring structure, then you're forming the ring between carbon number 1, 2, 3, 4, and the oxygen there, which is on carbon number 5. So that's where the ring system is forming. But because you can have this open chain form, and because that aldehyde group is there, that aldehyde can be oxidized, and so the whole sugar is known as a reducing sugar. Okay? So other ways of describing that sugar is because you've got 6 carbons in the chain there, you could describe that as a hexose. And because there's this functional group there at the top there, which is an aldehyde from organic chemistry, you could also describe that as an aldose. Okay? So those are different ways of describing this glucose molecule. It's a hexose, there's 6 carbons in the chain, or it's an aldose, there's this aldehyde group up here. And that open chain form is not the predominant form in solution. The predominant form is the cyclic form, like that. And for alpha glucose, you've got this arrangement. Excuse my drawing. Hope you can see what I mean. And you've got the CH2OH there. So if you count round here, 1, 2, 3, 4, 5, you can see that linkage there through the oxygen atom between carbon 1 and carbon 5 is exactly what we had in the straight chain form here. And the reason I've drawn this out here is because when you're coming down to this test here, boiling with the Benedict solution, that's sometimes described, isn't it, as a test for reducing sugars. And these sugars are reducing because they have an aldehyde group here, which itself can be oxidized. Now, so that's this point about the use of Benedict solution to determine the presence of a reducing sugar. But here there's another step where you test with Benedict solution for your reducing sugar after acid hydrolysis. And that is a clue for looking at this molecule here, sucrose. Glucose is a monosaccharide, isn't it? Right? Just one of these ring forms. Sucrose is a disaccharide. So that means 2 of, not 2 glucose units, but 2 single of these monosaccharides joined together. So sucrose is a 2 monosaccharides joined together. It's glucose and fructose. Okay, you can hear the voiceover, even if the writing is extremely poor. Sorry about that. Okay, so fructose, if we drew the straight chain form of fructose, again, fructose is a hexose. All right, so that means 6 carbons in the chain, just as we had with glucose over here. But in addition to being a hexose, it's not an aldose because it doesn't have an aldehyde group up here. That's an alcohol group at the end there. And then one step down, you've got a ketone group there. So in addition to being a hexose, just as the glucose over here was a hexose, glucose was an aldose because it had an aldehyde group. Fructose is a ketose because it has one of these ketone groups. And then you've got alcohol groups on the way down. And again, you're going to get the ring system starting from, this is carbon 1, 2, 3, 4, 5, 6. Again, the ring system is from the 5, but this time it's going round to the ketone on carbon number 2 rather than going to the aldehyde on carbon number 1. And so instead of ending up with a 6-membered ring, as you do with glucose, you end up with a 5-membered ring, like this. And you have your numbering then starts here, 1, 2, 3, 4, 5, and then you've got another CH2OH group up there. And you can see here, when I drew the open chain form, there's a, we're going from carbon number 2 through the oxygen to the carbon at number 5. And you can see here we're going from the carbon at number 2 through the oxygen to carbon number 5. And the, the point that I'm trying to make is that sucrose is a disaccharide between a glucose monosaccharide and a fructose monosaccharide. And both of those are reducing sugars, but when they're joined together, then these ketone and aldehyde groups are unavailable. So that means that sucrose is not a reducing sugar. But if you split it up into its constituent monosaccharides, and that splitting up process is called hydrolysis, then once you split it up, then the constituent monosaccharides themselves are reducing sugars. And so this step here, well, you test for your reducing sugar with Benedict's after you've run the hydrolysis. And the hydrolysis is splitting up the disaccharide into those two monosaccharides, which are both reducing sugars. So that's the distinction between those. The biuret is your test for protein, and iodine, of course, is your test for starch. Okay, so you've then got to look through. I think when I first saw that, I thought that was quite a confusing table. But you've got to look through this table and work out where this combination of starch and sucrose and glucose and protein can fit in. And if you run through that table, you get to answer C. Let's just check that. So if we added iodine solution, we should be detecting starch in solution number 1. So solution number one can contain starch. Okay, got it. And then here we're boiling with Benedict's, so we should be detecting a reducing sugar in solution number 2. So solution number 2 contains glucose. That fits. And then this step here, that we do acid hydrolysis first, and then you test for the reducing sugar. So that would test for sucrose, wouldn't it? So that would be in solution number 1, which it is. And then finally, we're testing for protein here. And so that would mean there should be a protein in solution number 2. Solution number 2 is protein. Okay, so that's just a sort of quick run through your test for biological molecules, and the reason why you get these positive results with Benedict's solution, because you're testing for, in the case of glucose, this aldehyde group, and why you get this positive result with sucrose only after you've done the acid hydrolysis, because you split up the disaccharide into the two monosaccharides. The disaccharide is not going to give a positive result with Benedict's, but each of the monosaccharides will. Okay, so still going strong now with these biological molecules. These are 2 monosaccharides. This is the one we just looked at. This is the alpha glucose. This is beta glucose, and you can see the orientation of that alcohol group is different in the alpha glucose and the beta glucose. And you've then got to match up these polysaccharides and say which monomers are used in the building up of those polysaccharides. So cellulose is polymer of beta glucose. Yeah? And when the glycosidic links form between these monomers, you get a chain where they're flipped 180 degrees as you go along the chain. So that they haven't asked for that in this question, but you sometimes get that asked in exams about do you get a 180-degree flip between each successive monomer in the polymer chain. So cellulose is a polysaccharide, a polymer built up from beta-glucose units. And then all these three are all polymers built from alpha-glucose. Okay, so the beta-glucose is going to that one, and then the alpha-glucose is used as a monomer for each of these three. So while we're here, we just distinguish between them. So those two, remember, are found in starch. So that gives us some discrimination. And then this one, in addition to having the 1,4 glycosidic links, has some branching. There's no branching there. There's a straight chain. This is branching. And the branching happens through this alcohol group here. So that you end up with 1,6 branching. So the polymerization happens between 1 in one of the molecules and the number 6 in another molecule. So you get 1,6 branching here, and these are relatively short chains. And that distinguishes the amylopectin from glycogen, where you get more 1,6 branching and you get longer chains. So that's the way that you distinguish between each of those different polysaccharides. The cellulose is from beta-glucose. All the others use alpha-glucose as the monomer. The amylose is a straight chain. The amylopectin has 1,6 branching, but is quite short. And the glycogen has much more 1,6 branching and has longer chains. So, well, let me get back to the question. So which monomer, okay, it's fairly easy actually. So we went overboard a bit there with looking at each of those polysaccharides. So the response here was, I've got down as B. Let's just check that. So the beta-glucose is going there. That's X, isn't it? And then you've got alpha-glucose, alpha-glucose, alpha-glucose. Okay, which is what we had. Good. Okay, so carrying on now with some more questions about biological molecules. And we've got an unknown, unfamiliar polysaccharide here, again linked by 1,4 glycosidic bonds. And it's similar in structure to glucose. And the key here is that in these long chains, there are hydrogen bonds between adjacent chains. Okay, so and the one that fits most closely to that is cellulose, which has both hydrogen bonding within the chain and between the chains. And so because you've got hydrogen bonding between chains, then that's a fit with cellulose, which also has hydrogen bonding between chains. Okay, this next one, continuing with structures of biological molecules. So a triglyceride, remember that the structure of that is that you have, I'm drawing a skeletal formula here, you have a 3-carbon molecule with 3 alcohol groups in it. So if you're in a chemistry class, that would be called 1, 2, 3 propan triol, because in chemistry, prop means 3, because you've got 1, 2, 3 carbons. It's a triol, there's 3 alcohol groups, and they're on successive carbons, 1, 2, 3. In biology, you have more familiar names rather than systematic names, and the familiar name for that is glycerol. And in the mark scheme, the examiner said that a lot of students accidentally chose answer A because they mixed up thinking of the word glycerol with the word glycogen. Okay, you just got the GLYC at the beginning, I suppose, that makes you think that they're, or get them confused. So glycogen, as we just saw, is a polysaccharide, and glycerol is just this small triol molecule. And to make your triglyceride, then you have in chemistry, this would be called a carboxylic acid. And then there's the carboxylic acid functional group, and then there's a long chain of long carbohydrate chains. Sometimes that's unsaturated and you get some double bonds in there. And so you end up with what's called an ester link forming between the alcohol group that's there and that part of the carboxylic acid there. And just rub all that out. So you end up with and that's the long carbon chain there, isn't it? Excuse me, didn't get my pen to quite right work there. I think you know what I mean, even if it's not drawn very well. Okay, so, and those are all called esters, that linkage there, and that's the long carbon chain. And so that fatty acid is now linked to the propan 1, 2, 3 triol, or the glycerol, through an ester link. And that combination of 3 fatty acids through ester links to the glycerol, that's your triglyceride. Okay? And occasionally you end up with just 2 of the carbon chains there, and instead you end up with a phosphate group, don't you, at that point? And then that's phospholipid. Okay, so those are the biological molecules that are based on glycerol, either 3 fatty acids and you get a triglyceride, or 2 fatty acids through the ester linkage and a single phosphate group, and you've got a phospholipid. Okay, so that's the structures we're working with here, and which statement about triglycerides is correct? So, excuse my bad writing there. Well, if we discounted that one, that was just a confusion between glycerol and glycogen. And this, you see, what they're doing, they're kind of teasing you with words in case your understanding is shallow rather than deep, because you know saturation is something to do with these triglycerides as well. But the saturation is referring to the double carbon-carbon double bonds or the fully saturated carbon-carbon single bond. So you might have in there a carbon-carbon double bond which is unsaturated, or a carbon-carbon single bond which is saturated. And so they're mixing up this idea of saturation with phospholipids, and there's nothing at all related between saturation and phospholipids. But you know, these things might be weakly understood in your mind, and so that's a kind of distractor here in this question in case you don't have a deep understanding of the structure of these triglycerides. And again, this is kind of playing with any weak understanding that you've got. The bilayer in the cell surface membranes, of course, is nothing to do with triglyceride, but is to do with that phospholipid, where one of the fatty acids is removed and you replace it instead with a phosphate group. So that's wrong, isn't it? And that's wrong. So by the process of elimination, the only one that's left is D. And if we wanted to positively identify it, as well as negatively exclude it through a process of elimination, well, we've been talking earlier, haven't we, about carbohydrates. And you can see in these carbohydrates, which are built from these monosaccharide monomers, there is a high proportion of oxygen. There's one in the ring system there, and there's these alcohol groups all the way around the ring. Those polysaccharides built up from monosaccharides, well, they have a high proportion of oxygen, don't they? When you come down to look at these triglycerides, well, there's some oxygen at this glycerol end of the molecule in these ester linkages, but you might have something like C17, like a really long chain here. And all that chain is just carbon and hydrogen, no oxygen at all. And so it's true that these triglycerides do have a much lower ratio of oxygen to carbon than the carbohydrates. So we can either identify the answer there positively, because we've got a good understanding and we can compare it back to the carbohydrates, or we can negatively exclude it through a process of elimination, because all of these are just assuming that you've got a very shallow understanding of triglycerides rather than a deep understanding of the fatty acids joined through to the glycerol through the ester linkages. Okay, well, I think this paper is showing you that you have to have a good understanding of size in your head. We had all those questions on magnification and the size of molecules and biological entities earlier, but we've been through a whole bunch of questions here which are all to do with biological molecules, and that's continuing now with this question about amino acids. And actually, I think this one, that's a pretty easy question to get, because amino acids all have, don't they, the same basic structure, which is you've got a carbon in the middle, and then you've got a carboxylic acid, and then you've got an amino group. Okay, so you've got amino group, carbon, carboxylic acid, and then you've got a hydrogen, and then you've got all the different side chains that give you the 20 different amino acids. But the common, or the commonality between all the amino acids is that amine there, and the carboxylic acid there, and the carbon in the middle there, and the various side chains that give you the different amino acids. So when you look at these ones here, well, that's not too difficult, is it? Because you're getting the amine, and then the carbon and the hydrogen and the carboxylic acid. And you've got the amine, the carbon and the hydrogen, and the carboxylic acid. And so that amine there is just part of one of these side chains, and that carboxylic acid there is a part of one of the side chains as well. So it's that part up there that is the common part of all the different amino acids. And the different amino acids are distinguished between the different side chains that they have. Okay? So when the peptide bond forms, because you're joining one amino acid to the next, well, if we drew them, that's the next amino acid, and it's got its carboxylic acid group there, it's got its amine group there, and it has whatever its side chain is there. I'll just call that R dash to make it different from that. And so the peptide bond is going to be forming from the condensation reaction between the carboxylic acid group of one amino acid and the amine group of another amino acid. And when those react together in the condensation reaction, you're going to get that's from the carboxylic acid group, and that's from the amino group, and then that goes off to the rest of the amino acid. And that collection of atoms there, that's a carbon in there, okay, which is from the carboxylic acid. That's that came from the carboxylic acid, and that came from the amine group there. That's the peptide bond. And so, which are the groups that are going to form the peptide bond? Well, it's going to be that joining with that. I mean, the way they've drawn this, it would be much easier to say, well, that carboxylic acid joining with that amine. But I suppose they're just kind of getting you to sort of think a little bit more carefully about that. So the groups that are going to join together to form the peptide bond, well, that's going to be 1 and 3. So that's answer A. Okay, good. So, and actually, this next one here is still really talking about these peptide bonds, because you formed in one peptide link that group of atoms, and we saw just now that that continues in the peptide chain, but that's the peptide link. And of course, these peptide chains can be thousands of amino acids long. I suppose more typically more like hundreds of amino acids long. But there's hundreds of amino acids in the peptide chain. And as these chains fold, then the chances are that the oxygen from one peptide link can come into proximity with the amine of another peptide link. So that's another, I'm getting into the text up here. So that's the amine, like assuming that the whole peptide chain kind of curled round, then you can end up with an amine coming, sorry, that NH group there coming into close proximity with that group there. And once you do that, then there's the chance that you get a hydrogen bond forming between the N, the H, and the O there. All right. Now, so if I just draw that up, so you're getting, in chemistry, that's the carbonyl group, and this in chemistry, that's an amide group. So you're getting a hydrogen bond there, and that's the amide group. Okay, that's the hydrogen bond. Okay, and that hydrogen bonding between the carbonyl group in one peptide and the amide group in another peptide link, that's the basis of secondary structure in proteins. And that secondary structure can either be an alpha helix or a beta sheet. Now, the one that I've drawn up here is probably closer to the alpha helix. So if that's the protein chain, then what's happening is you've got a carbonyl group from one peptide bond, and you're getting, my drawing's a bit off here, you're getting the NH from another peptide link, and you get the hydrogen bond between them. And that set of hydrogen bonding, if I just drew it in like this, is what's holding that alpha helix in its shape. So that's the origin then of the secondary structure in proteins. There's the alpha helix, and you can get, if that's the protein chain, and the protein chain is there, that's you can get hydrogen bonding forming between these two peptide chains, and that's the beaded sorry, beta sheet. It used to be called beta pleated sheet. The beta sheet between the two polypeptide chains. But this hydrogen bonding between the carbonyl group and the hydrogen of an amide group is the secondary structure for proteins. Now, for each of these proteins, like protein subunits, I think here, you can see that's the continuing protein chain, and it just ends up there. And these blocks here, what they really mean by that is that that chain then continues like this in an alpha helix, because you're getting those hydrogen bonds forming, and the protein chain continues. And that's what, you know, the diagram. These tubes are intended to show this alpha helix here. So, so the primary structure is wrong, isn't it? Because the primary structure is the order of amino acids. So whatever amino acid there, defined by its side chain, is joined to another amino acid. The order of which amino acid is joined to which is the primary structure. So it's nothing to do with primary structure. And so it's definitely C or D. And again, really, this is just testing your, you know, how kind of shallow your thinking might be, because the peptide bonds, of course, are all, the covalent bonding within the peptide bonds is all related to primary structure, isn't it? That a peptide bond formed because it joined one amino acid to another amino acid. And so the secondary structure is held in shape by these hydrogen bonds. We saw that there between these loops of this alpha helix. And so the answer for that then is answer C. It's a secondary structure held in its shape by hydrogen bonds. All right, so in some ways, the papers seem to follow almost a kind of logical flow, because we started off by looking at sizes and going right down to actually the size of molecules. And then it's moved on to joining those individual molecules into biological polymers. We've looked at polysaccharides, and we've looked at polypeptides, proteins, and we've looked at triglycerides. And so once we started to talk about chains of amino acids, then the large chains of amino acids are forming proteins. And so the next set of questions start to talk about proteins and talking about actually enzymes and enzyme kinetics. Okay, so again, this is taking your knowledge of lock and key and induced fit theory into an enzyme that you're probably not familiar with. Doesn't really matter. Tells you a little bit about that particular enzyme, and it's talking about induced fit. So the idea here is that if you've got an enzyme which is made from a chain of amino acids, then there might be a kind of pocket in the enzyme. And the original theory was that the substrate that the enzyme is going to act on is a perfect fit for the substrate. So that's a so-called lock-key hypothesis, that the substrate perfectly fits in to this pocket within the enzyme. A more sort of nuanced version of that is induced fit, where as the substrate binds to the enzyme, then the enzyme changes its shape so that it becomes a good fit for the substrate. So the final shape of the active site of the enzyme is induced by the shape of the substrate. So it's a little bit more sophisticated than just thinking about a lock and a key, the induced fit model. So that excludes, doesn't it, number one, because the induced fit model, in the induced fit model, the shape doesn't have to be complementary to begin with. The shape becomes complementary when it's induced. The active site fits the substrate while the substrate is bound to the active site. So that first description up there is part of the lock and key hypothesis, not the induced fit hypothesis. So we can exclude that one. So non-competitive inhibitors, those are inhibitors that bind to somewhere else in the protein chain and then distort the entire enzyme. And so whether you've got an induced fit model or a lock key model, if something is binding to the enzyme and is distorting the whole enzyme, then that's going to affect the enzyme, whether it's induced fit or lock and key. So that doesn't form one of the answers to the question either. But it's true that either the lock and key mechanism or the induced fit mechanism, once the substrate is in the active site and the active site has properly bound to the substrate, then these various groups that are individual for each of the amino acids, those various groups can then bind to the substrate and convert the substrate into the product. So whether you're working with a lock and key mechanism or whether you're working with an induced fit mechanism, then it's true that these amino acid side chains are the ones that start to catalyze the conversion of the substrate into the product. So for 12 here, then only 3 is the answer. Okay, so now question 12 was about non-competitive inhibitors. Competitive inhibitors, then, are going to be competing for the active site with the substrate. So a competitive inhibitor will have a similar shape to the substrate. It's going to be competing for the active site. So competitive inhibitors do not bind to a site other than the active site. The competitive inhibitors are competing for the active site. So by the process of elimination, it's got to be C or D. It can't be A and B because competitive inhibitors aren't binding to some other part of the enzyme molecule. Now, and again, this is kind of just testing, because for some students, if you don't have a deep understanding, you'll just recognize some of these words, like we had earlier, and you'll recognize the word without having a deep understanding of the use of the word within the particular context. And so you may know that catalytic reactions work by lowering the activation energy needed for a reaction to occur. So activation energy and lowering activation energy is certainly part of the way an enzyme catalyzes a reaction, but it's nothing at all to do with the competitive inhibitors. And so you're getting 2 crosses there to give you answer D. The competitive inhibitor has a similar size and a similar shape to the substrate, so it can compete for the binding of the active site. Okay, more enzyme kinetics here. Okay. So so this is just the enzyme reacting with substrate all on its own, that one there. This one here is working with the competitive inhibitor, and this is working with the non-competitive inhibitor here. And what's happening at point 1 is that you can see as you increase the concentration of substrate, the rate of reaction goes up. So that must mean that there is plenty of enzyme available. And so what's limiting the rate of reaction there is substrate concentration, because as you increase substrate concentration, the rate of reaction increases. So at that point, substrate concentration is the limiting reaction, because you can see as you increase substrate concentration, rate of reaction increases. Eventually, as you keep adding more and more and more substrate, you've saturated all active sites. And no matter how much more substrate you add, it doesn't matter, because all the active sites are already full with substrate. And you can add as much substrate as you like, you still can't fit any more substrate in because all the active sites are full. So at that point, the enzymes are saturated, all the active sites are saturated, and so the limiting factor at that point is enzyme concentration. Now, at this point here, the reason that the rate is lower, of course, is because you're using a competitive inhibitor. So at that point there, the competitive is stopping the substrate binding to the active site. And in 4, the non-competitive inhibitor binds to the enzyme, it distorts the shape of the enzyme and reduces its catalytic activity. And so no matter how much substrate you add, you can't get away from the fact that the non-competitive inhibitor has distorted the shape of the enzyme and reduced its catalytic activity. And so your answer there is, well, at number 1, then you don't have enough substrate. If you add more substrate, the rate of reaction increases. At number 2, all the active sites are saturated, and to increase the rate of reaction, you need to increase the enzyme concentration. In number 3, the rate of reaction is reduced because the competitive inhibitor is taking up some of the active sites where the substrate needs to be to form product. And in 4, no matter how much substrate you add, it's not going to increase the catalytic activity because the non-competitive inhibitor has distorted the shape of the enzyme, reducing the catalytic activity of the enzyme. Okay, the clue to number 15, I think, was this here that tells you that is a cell surface protein. And because you've got a cell surface protein there, then that is the outside of the cell, that is the inside of the cell. That's the kind of way that you get into this question, I think. And so you know that if there's respiration occurring inside the cell, you have a high concentration of carbon dioxide inside the cell, and then carbon dioxide would need to move out through diffusion . So number 2 then is, you see carbon dioxide is moving out through diffusion. Okay, we had that carbon dioxide going up, sorry, that's CO2 there, isn't it? Carbon dioxide moving out of the cell here. Okay, so that's the way you also unpick this question a little bit. You recognize there's a cell surface protein, that's the outside of the cell, that's the inside of the cell. You've got things diffusing out. Well, you've probably got a high concentration of carbon dioxide from respiration inside the cell, that's diffusing out. So that's the way that you sort of unlock that question there. Okay, just wanted to talk a little bit about the nucleic acid molecules. Okay, so one of the things you've got to learn for nucleic acid molecules is not only the bonding that you get. For example, you know there are 4 bases in DNA, and you get 2 hydrogen bonds between adenine and thymine, and you get 3 hydrogen bonds between cytosine and guanine. But you've also got to know, haven't you, the whether these are derived from purine or pyrimidine rings. So the purine rings have nitrogen there, nitrogen there. You have bonding here, here, here. So those are pyrimidine rings. And then the purine rings have a 5-membered ring. And a 6-membered ring. Yeah, sorry about my drawing. Okay, all right. So you've got the 2 basic ring systems. Pyrimidine is the smaller one. Purine is the larger one. You've got 2 rings together there. And you need to know that the cytosine, the thymine, and then the uracil in messenger RNA and transfer RNA is based on the pyrimidine system, and then your adenine and your guanine is based on the purine system. All right, so with all that in, that's all something you have to learn because it's getting tested in this question here. So, well, we've got something that's joining to adenine. Well, what joins to adenine in DNA is thymine. So that means that it's going to be that one, isn't it? Or that one down here. So the base is thymine, and it's joining with 2 of these hydrogen bonds. And the thymine is based on pyrimidine. So pyrimidine. So the only one that fits then is you've got T, thymine, that bonds with A, adenine, with 2 hydrogen bonds, and the thymine is based on this smaller ring system, pyrimidine. Okay, so it's not sufficient to know the 4 bases in DNA or the 4 bases in RNA, or even to know the bonding between them, 2 bonds or 3 bonds. You've also got to know the basis of these bases, either pyrimidine or purine. Okay, so in this next section here, they are working with your understanding of transcription and translation, and with your understanding of triplets in genetic code, and then codons in messenger RNA, and anticodons in transfer RNA. So you need to have that all quite sorted out. But you know that the genetic code is a code of 3 letters, all right? So you've got a genetic code triplet there, you've got another triplet there, you've got another triplet there, haven't you, and another triplet there, okay? And this is a nucleotide sequence of a gene, okay? So this is talking about genetic code and triplets. Okay, and then you've got to think about the transcription of that code into codons for messenger RNA. My goodness, my writing. Okay, so you're looking for the transcription of these genetic code triplets into codons in messenger RNA. Okay, so, well, you know that the C is going to have a complementary base of a G. C, G. And then you've got G, C, G. Now, the complementary base pairing in DNA, of course, is A to a T, but the complementary base pairing between the genetic code and the messenger RNA is you need a uracil there. So C has a complementary base of a G, complementary base of a C, complementary base of a G, C, G. Now, my writing is not very good there, I'm afraid. But what you end up with then is you then take these codons and you have to look them up in the genetic dictionary of our messenger RNA. This is the messenger RNA genetic dictionary. So if you look up those codons, I had CGC, quite a lot, didn't I? There, CGC is the amino acid alanine. So CGC, and then you've got sorry, GCG, 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 and then you've got UGC, which is that one there, isn't it? And then you're back to GCG, which is that one there again. So that ends up as being the codons in the genetic dictionary for messenger RNA of alanine, alanine, cysteine, alanine, which is A. Okay, so they're still working with you here about nucleic acids. And the first one is that it's true that there is some nucleic acids that carry an amino acid. Now, the one that you're thinking of there, of course, is transfer RNA. Yeah, it's a small piece of nucleic acid, but it has this kind of structure, doesn't it? Where you've got these kind of little pieces there. And then at this end of the transfer RNA, you can bind on a particular amino acid, and you have a particular codon here. So it's true that transfer RNA can carry an amino acid. So that's true. Now, the genetic code, if we go back up here, the genetic code is in the gene inside the nucleus, isn't it? So the genetic code is there and it doesn't leave the nucleus. The thing that leaves the nucleus is the transcribed messenger RNA with its codons. That's what leaves the nucleus. So it's not true that the genetic code can leave the nucleus. Only the transcribed genetic code in the codons of the messenger RNA can leave the nucleus. And the same way, the genetic code can't be carried to a ribosome, can it? The genetic code that's been transcribed as codons in messenger RNA, that's the nucleic acid that can move into the ribosome. And so that's wrong, that's wrong. The amino acids are held in position for translation. That's true because a number of these transfer RNAs will bind to the messenger RNA in the ribosome, and then there'll be a condensation reaction between consecutive amino acids to join them together. So it's true that amino acid can be held in position by a nucleic acid. And so for that one, 1 and 4 are correct, which gives you your answer B. Okay. So just scanning through the last of these questions here, the examiner picked up that in question 25, a lot of students, strangely, had very weak understanding of transpiration. So you had to come here with the correct definition of transpiration, and the examiner was saying that only very, very able students, for some reason, had a correct definition of transpiration. So you need to know that transpiration, first of all, is evaporation, and then diffusion in the airspace inside a leaf. Okay, so this transpiration is first the evaporation of water, and then once that water is evaporated to become water vapor, it has to diffuse out of the leaf through the stomata, and that process is transpiration. So if you look through there, which one is it? Can't see it at the moment. That one was C, the loss of water vapor through these air spaces inside the leaf out of the atmosphere. And the examiner flagged up that that was one of the ones the students were generally weak on. Okay, I think the last one from me then, what is the view of the heart here? So quite often you draw simplified diagrams of the heart. This is a much more realistic diagram. P up here, you can see it's actually the larger, thicker tube is the aorta. Q is the pulmonary artery. R is the pulmonary vein. And S is then the vena cava here. And so that one has oxygenated blood going out, doesn't it? This has oxygenated blood coming back in. I can get a different paint color here. This is deoxygenated blood going out to the lungs. And this is deoxygenated blood from the body coming back to the heart. And so you had to say, didn't you, out of all of these, which one was the correct answer here? If we look through them, the only one that fits then is B, because P and R is carrying blood that is saturated with oxygen. Well, P is carrying blood saturated with oxygen because that's leaving along the aorta here, and R is saturated with oxygen because this is blood returning from the lungs back to the heart. So that's oxygenated blood coming back into the heart. And so the only answer there is B. But it did rely on you having much more than a kind of simplified version in your mind of the heart, to actually having a biologically realistic version of the heart, and being able to identify the aorta leading back to the body, the pulmonary artery leading to the vein, the pulmonary vein coming back from the lungs, and the vena cava, which is bringing the deoxygenated blood from the body to the heart. Okay, so I think that's where I'm going to stop for this masterclass. Some of the questions later on about disease, I think, were fairly straightforward. There were some diseases which were clearly, like this one here, some of these were infectious diseases. Others of them were genetic diseases, like the sickle cell anemia here is a genetic disease, nothing to do with an infectious disease becoming more susceptible through a suppressed immune system. Similarly, these kind of diseases here, the HIV, AIDS is treated because you can treat that with nucleotides. Similarly, some questions about rabies again there. You just had to understand the kind of disease and the way it was treated. Okay, so that's where I'm going to finish there with this masterclass. I think the main points that I wanted to bring out was how important it was in your mind to have a good understanding of the sizes of different molecules, because and biological entities as well, because that occurs typically again and again and again in the first parts of these papers. You can see then almost a quarter of this paper was on biological molecules. So even though that may only form one or two maybe chapters in the revision notes or in the book that you're using, a substantial part of the paper assumes you have very good understanding of carboxylic acids, proteins, triglycerides, nucleic acids, primary structure, tertiary structure, secondary structure, proteins, quaternary structure, that's all tested. So all the structures of biological monomers and macromolecules is tested pretty thoroughly. Then the bits on enzyme kinetics as well. So I think the things that I've been emphasizing in this paper have very much been your work on biological molecules and on the relative size of the different biological entities that you're working through in the studies of biology that you're doing. Okay, so that's the end of the masterclass this evening. If you wanted to follow up with any of the Z coaches, they're on the Slack channels, and of course, extremely very welcome to use the Slack channels, speak to the Z coaches that are there. And that just remains for me to wish you every success in your studies of biology for Cambridge International exams, and welcome you to the next biology masterclass when that's on the next occasion when that's run from Cambridge Leadership College. Okay, thank you very much, and look forward to speaking with you next time in the next masterclass.